What is CRISPR? CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. This is a mouthful, so let's break it down. The second part of the acronym, Short Palindromic Repeats, or SPR in the word CRISPR, simply says there are short segments of DNA, usually 20 to 40 base pairs. These 20 to 40 base pairs are palindromes, meaning they are read the same forwards or backwards. These short segments are all identical and repeat throughout the DNA sequence. Next, clustered regularly interspaced, or CRI in CRISPR, means the identical DNA segments are interspaced in the DNA segment, or there is a space between each segment. So, between each identical component, there is spacer DNA, an essential piece. A spacer DNA is similar to the DNA of a bacteriophage. It identifies with viral DNA.
This CRISPR system was first discovered in the bacteria E. coli and functions as a bacterial immune system. The spacer DNA that is incorporated is not identical, rather each segment is unique. These unique segments match up perfectly to viral, especially bacteriophage DNA, allowing the bacteria to have immunity to certain pathogens. Also discovered within the system was CRISPR-associated genes, otherwise known as Cas genes. Cas genes code for Cas proteins, which are helicases and nucleases. Helicases unwind DNA, while nucleases cut the DNA. When a virus infects a bacterial cell, injecting its DNA into the cell, the bacteria transcribes and translates the DNA to create Cas proteins, as well as transcribing the specific spacer DNA that is identical to the pathogen's DNA. The result is CRISPR RNA, or cRNA. The CRISPR RNA fits right into the Cas proteins to create a complex. The bacteria can now use helicases and nucleases to cut and deactivate the bacteriophage DNA.
What if there is no identical segment of spacer DNA for a certain bacteriophage? In this case, a different protein will be transcribed and translated to deactivate and incorporate the new DNA. The viral DNA is copied and then included into the CRISPR system, so the next time this pathogen attacks, the cell can fight it off quickly. So how can we use this in humans? We needed to figure out how to hijack the system to cut specific DNA sequences in humans, rather than in viruses or bacteria. gRNA, or guide RNA, is a lab-made molecule containing the specific DNA sequence that needs editing.
In humans, we take a piece of DNA that needs editing, also called the target DNA. Then, we manufacture a complementary gRNA sequence in a lab to match the target DNA. Cas9 is a type of Cas protein that creates a complex with the gRNA. The target DNA will feed into the Cas9 complex, cutting the DNA at these 2 points. This results in 2 genes which the cell will try to repair using non-homologous end joining. This is just a DNA repair mechanism that fills the gap with random nucleotides, therefore inactivating the entire DNA segment. However, we want to knock out these bad genes and insert good genes. There are now 3 parts to the system: Cas9, gRNA, and host RNA. The host RNA is the sequence that we want to insert into the DNA. The host RNA is inserted and the complementary nucleotides are attached. This is accomplished through HDR, or homology-directed recombination. We have now directed the insertion of nucleotides to create a functioning gene, or knocking in a good gene.